Hello everyone, welcome back to our discussion on the biotechnology or the Bio 134. As a sort of review, last time we discussed on how to genetically modify the organisms, so the steps involved in modifying the organisms. So let us have a fast look on the topic we have discussed last time. There is a steps to genetically modify an organism. To genetically modify organisms, we need to have a vector, and the vector should be in the form of plasmid, by which a plasmid is fragments that has been obtained from a bacteria. Then the plasmid has been collected, then inserted with a desired gene collected from other organism, and after that uh, desired gene has been inserted in the plasmid, then it is form part of the plasmid. Then uh, the engineered plasmid inserted into the new organism, into the new bacterium. Then it will incorporate and associate in the systems of that particular organism. Then that particular plasmid carrying desired genes will replicate following the replication process of the receiving or the host cell. So this is the more or less the basic steps. Now isolating the genomic DNA, then fragmenting the DNA, screening the DNA fragments, the insertions of the DNA in a vector or the plasmid, then introducing the DNA in a host with the plasmid, then culturing the cells, then transformations of the host cells. This is now the genetically modified organisms. No? All right. Now, to continue our topic today, so let us start with a discussion on the tools of recombinant DNA technology. Okay. Let me enlarge this now. All right. Let me use my pen. All right. So we have some tools of recombinant DNA technology. Yes, as we all know that when we say recombinant DNA technology, we're talking about the new or the host cells carrying the desired gene that has been inserted into the plasmid. Then that plasmid carrying the desired gene will be inserted into host cells, so that the desired gene that has been inserted into the host cells will replicate following the replications of the host cells. Okay, what are the different tools in the recombinant DNA technology? So first, recombinant DNA technology is accomplished with tools. No, one of the tools is the restriction enzymes. Now, from the word restriction enzyme, it is an enzyme that will cut a certain portion of the DNA strand, and taking and obtaining the desired genes out from that DNA strand. All right. So restriction enzyme, uh, we have the restriction endonuclease, and we have the restriction exonuclease. Now, we have the two types of restriction enzymes. We have the restriction endonuclease and the restrictions exonuclease. And another tool, we have the polymerase enzymes. No? And we also have number three, we have the ligase enzymes, the vectors, and we have the host. So using these different tools, we'll be able to perform or conduct DNA, recombinant DNA, or we are able to modify the DNA of a particular organism. So take note for that. So we have the tools of restriction enzymes, polymerase enzymes, ligase, vectors, and host. Okay. So what are the restriction enzymes? Okay. Uh, Enzymes which is used to make cut in the DNA in recombinant DNA technology. So the purpose of the restriction enzyme is to cut a certain portions of DNA, particularly the desired genes. Yun ang kukunin natin, i-transfer natin later on sa host cells. No? So in 1963, two enzymes were isolated from Escherichia coli, which restricts the growth of bacteriophages. Okay, when we say bacteriophages, we're talking about the virus-eating bacteria. So bacteriophage is a virus-eating bacteria, and Escherichia coli or the E. coli per se. These are the bacteria usually found in our stool, in our feces, in our sa ating mga dumi, no, sa tae sa tao, sa tae sa animals. So mayroong mga Escherichia coli diyan. But it was found out that there are enzymes that can be derived from E. coli that restricts the growth of bacteriophages. Okay, so one enzyme added methyl group to DNA, other cut DNA, and was named restriction in the nucleus. Again, one enzyme added methyl group to the DNA. So in the restriction enzyme, so added methyl group to a DNA, and other cut the DNA and was named as restriction endonuclease. So again, so restriction endonuclease, this enzyme added methyl group to the DNA and other cut DNA and was named as restriction endonuclease. When we say methyl group, we're talking about here the group of alcohol. Methyl group, the if you have your organic chemistry, if you have your biochemistry, if you can still recall, this is the methyl group actually is the CH3 functional group in biochemistry, no, the methyl group. Anyway, all right. So we have the restriction endonuclease. So the functions of restriction endonuclease depend on a specific DNA nucleotide sequence. When we say DNA nucleotide sequence, we're talking about the different nucleotides. So there are different nucleotides in the DNA strand. So in the DNA, we have the double-stranded DNA, no? A double helix DNA. And along that strand of DNA, there are some groups of, excuse me, class. There are some groups of, what you say, purines, right? Purine helix. Napakay ba, pirmahan? Akong gisat ay dati lamok. Ano to lasa to ka? Window transaction. Okay, ha? Basta, kailangan natin umanda. Kinakat? Sige. Okay. Sige, proceed ako. Alright, so let us proceed. So we have the function of restriction in the nuclease depends largely on a specific nucleotide sequence. When we say a nucleotide sequence, we are talking about the sugar-based pairs, no, on the double-stranded DNA. In a strand of a DNA, there is a sequence of nucleotides. So we have the purine nucleotides, no. So I'd like to write in here. So we have the purine. Sorry, where's the? Okay, let me show my pointer. So we have the different nucleotides. We have the purine, no? In DNA, in a strand, there is a purine nucleotide, which is composed of adenine and guanine. Okay, in a DNA, and ito yung ang dalawang nucleotides ng purine, we have adenine and guanine. Okay, and we also have pyrimidines, base pairs. Now we have purine, we have also pyrimidine. So we have pyrimidine, pyrimidine. And we have two types of pyrimidine. We have the cytosol and thymine. So this is thymine, no? So again, in DNA strand, we have a double strand, and along that strand, there are some nucleotide sequence, a sequence of purine and pyrimidine. So a purine composed of adenine and guanine, and the pyrimidine composed of cytosol and uracil. This is for DNA. But in RNA, so ma-change doon ang pyrimidine. Ang pyrimidine becomes cytosol and uracil. Mawala doon ang thymine, no? So, yun po. So, klaro ko lang po, in a DNA sequence, there's a nucleotide sequence, and we have the different nucleotides. We have the purine and pyrimidine in the DNA. Sa DNA, ang purine is adenine and guanine. Again, sa DNA din, ang pyrimidine, meron tayong cytosol and thymine. But, meron tayong RNA. Doon sa RNA, ang purine, the same, meron tayong adenine and guanine. But doon sa pyrimidine, under sa RNA, we have the cytosol and uracil. And in the DNA-based sequence, in the pairing of nitrogen bases, so mga nitrogen bases kasi itong adenine, guanine, cytosol, and uracil. In pairing of that, so there is always a pair, opposite pair of, for example, in nucleotide of adenine, it is always pair with thymine. So ang purine na adenine, mayroon siyang pair doon sa strand na thymine. And kung ang strand naman mayroon siyang guanine, ang pair naman niya ngayon is cytosol. That is for a DNA. But kung doon sa RNA, sa RNA strand, ang pair ng adenine is thymine, but doon sa RNA, ang pair ng guanine is uracil. So, yun ang so, ito, isinot ko dito. For example, in an RNA, so, ang purine dyan, mayroon siyang, of course, adenine, and guanine. Pero, ang pyrimidine doon sa RNA, ang pyrimidine is magiging cytosol and uracil. Sorry, back tayo. Nawala to. But yun, no? Cytosol and uracil. So, nawala. Anyway, but let us proceed now. Okay, so yun, the functions of restriction in the nucleus depends on a specific DNA nucleotide sequence. Okay? The first endonuclease isolated was HIND2. No? HIND2, studies prove that HIND2 always cut DNA at particular point by recognizing a specific sequence of 6 base pairs. When we talk about base pairs, we're talking about adenine, guanine, cytosine, uracil. No? Again, the first endonuclease isolated was HIND2, and studies prove that HIND2 always cut DNA at a particular point by recognizing a specific sequence of 6 base pairs. So, recognition sequence of enzyme. So, this HIND2, so it will cut, no? It will cut the band between the double stranded band. For example, we have a DNA strand, so i-cut niya yun, no? Cut DNA at a particular point by recognizing a specific sequence of 6 base pairs. So there are 900 restriction enzymes isolated from 200 strains of bacteria. So na mga enzymes, na mga restriction enzymes. Yung mga enzymes na yon, mayroong specific portion, specific function as to what particular sequence dyan sa DNA strand na i-cut nila. Yan po. Okay? Okay. So still, no, in restriction enzymes, the restriction enzymes belongs to a nuclease class of enzymes. So nuclease class of enzyme. There are many types of enzymes, but the restriction enzymes belongs to a nuclease class of enzyme. It is of these two types. Now we have the restriction exonuclease, from the word exo, okay, removes nucleotides from the end of the DNA. So it removes nucleotides from the end of DNA. When we say nucleotides, again, we're talking about the adenine, guanine, cytosine, and thymine in the DNA, no? So may ikakat niya doon, no? Kung ano yung nasa tumoy sa DNA, so ang mag-cut doon is exonuclease, no? Ay cut niya is adenine, guanine, cytosine, yan ang mai-cut niya, no? Or thymine. Then we have another types of restriction enzyme. We have restriction endonuclease, no? So cut at specific position within the DNA. So sa tunga sila mo cut sa DNA from the word endo. Okay, take note for that. Okay? Para silang mga gunting, para silang mga scissors used to cut a certain portions of the DNA strand either on within or either on the end of the DNA, no? Okay. So while naming restriction enzyme, first letter represent a genomic name and second two letter represent specific name of the prokaryotic cell, no, from which the enzyme was isolated. Okay. So there is sequence of naming. For example, we have the examples of restriction enzyme. For example, E. coli. Okay, isolated from eco is Escherichia coli. The RY13, no? R, name of strain, and one, no, this is like eco R1, and one order in which the enzymes were isolated from that strain of bacteria. Okay, proceed now. So how restriction in the nucleus act? We're talking now about the restriction endonuclease. We're talking about an enzymes that would cut nucleotide within or at the center of the DNA strand. Okay. So RA, or the restriction in the nucleus, inspects the DNA sequence to find its specific recognition site to bind. So yung enzyme na yon, so parang siyang parang titingnan muna niya yung strand, DNA strand, inspect the sequence, tingnan niya kung saan siya pwede mag-bind. After that, tingnan niya kung saan siya pwede mag-bind and cut specific each of two strands of the double helix at the sugar phosphate backbone. Okay. So, inspect muna niya, bind, then cut. Okay? So, the restriction in the nucleus recognizes specific palindromic nucleotide sequence in DNA. When we say palindromic nucleotide sequence, we are talking about this sequence, the guanine, the adenine, thymine, cytosine. This is now the palindromic nucleotide sequence, and along that sequence, so these are the sequence that needs to be cut either at the center or at the end. Okay, this is the palindromic nucleotide sequence in DNA. And the palindrome in DNA, sequence of base pairs that reads the same on two strands when orientations of breeding is kept the same. Example, 5 to 3. When we read sa 5 to 3 in this first line, we have the guanine, adenine, thymine, cytosine. And on the other side, pag i-read natin dito, so the same din, no? The same din siya. Guanine, adenine, thymine, cytosine. Because, again, sa DNA, ang guanine is always paired with cytosine. Ang adenine is always paired with thymine. And, yun na, no? Ang guanine paired with cytosine. And, adenine is always paired with thymine. Therefore, in a double helix DNA, for example, it is one single strand and the other single strand. Itong dalawa na ito is double strand na ito, no? Kung na-remember niyo yung sequence ng DNA, ganun usually kasi eh. So, pag mayroong G dito, mayroong A dito. Pag mayroong A dito, mayroong G. Mayroong C dito. Ito, pag G, C ang partner. Pag A, T ang partner. Ang katulad niyan, mayroong A. So, T na naman. And, ilan may 1, 2, 3, mayroong T. So, A na naman. Then another T and A. This is the strand, no? Ito palang single strand ng DNA is must have an always equivalent complement strand of DNA. Ito pala sila mag-partner sila, but yung partner nila is based on pairing of guanine, cytosine, adenine, thymine. So, ganun po yun. So, this is the palindromic sequence. Tingnan po that. Okay. The restriction enzyme cut the strand away from the center. Okay? Pag sinabi, the restriction enzyme cut the strand away from the center. So, this is the restriction endonuclease. The restriction endonuclease cut the strand away from the center of a palindromic site, but between same two bases on opposite strand, leading to the formation of a single strand portion at the end, or the sticky ends. No? Sticky end, hydrogen bond with their complementary cut counterparts is the DNA ligase. By the way, itong adenine, for example, this is the double helix DNA duplex. Itong adenine na to pair with thymine, ang bonding between nila is hydrogen. There's a hydrogen bond. So, bakit kailangan hydrogen bond? Because hydrogen bond is a weaker bond compared to covalent bond. Kaya nga, madali lang natin i-replicate yung mga DNA strand because the bond between nucleotides is hydrogen bond. And hydrogen bond is a weak bond, allowing the bonds to be easily dissociated, easily cut, so that the DNA strand can be easily replicated. But kung covalent bond bang nandyan, so mahirap i-replicate yung mga DNA ngayon. But praise God, na ang bonding nila is hydrogen bond, now it could easily be duplicated, using the modifying the DNA. We can modify easily because it could easily be broken down ang bond nila and easily be replicated. Okay. So now, the sticky ends, hydrogen bond with their complementary cut counterparts. This is the DNA ligase. Now, sticky end. So, pag maputol itong mga bond nito, because of the DNA ligase enzyme. So, with the DNA ligase enzyme, yung bond na yan, ma-broken down, mahiwalay yung dalawang strand into individual strand, which later on that individual strand form their counterpart strand, forming now, replicating now, forming now a new DNA strand. Okay. For example, so this is an example of the DNA strand, no? Example of that. Then we have the restriction enzyme. Okay? Now, the restriction enzyme, gikat niya dito sa dito, no? Gikat niya itong double strand DNA na to, cut niya from this portion, no? Dito na portion, dito, cut niya. Okay? Cut, then sa kabila din, cut niya dito sa now, pag cut niya, meron ng open ends. Kung mayroong open ends, eventually, itong T, so partneran naman ito ng A, no, the adenine. And then, so mayroon na tayong new DNA, no? So, ito yung sticky ends na sinabi nila. Okay? For that. Let's proceed. So, we have different types of restriction enzymes. And examples of that is, we have the ECOR1, no? The E. coli. So, i-cut niya dito, portion na dito. At pag-cut niya doon na portion, cut din niya ang portion na dito. Cut. So, ito yun ang resulting, parang tayong overhang, no? Na mga overhang na DNA. So, mayroon din tayong PST1, no? The Providencia, Stuartii. These are bacteria, no? These are bacteria ito. Ito, bacteria din ito. We have the SMA1, the Serratia, Marcescens, no? We have the Hy3, the Haemophilus, Aegyptius. These are bacteria from which we have to obtain an enzymes, no, the restriction enzyme. These are just examples of enzyme which cuts a specific site in the DNA. For example, dito, no, ito siya, i-cut niya dito, itong Serratia marcescens. Ito namang Haemophilus Aegyptius, so i-cut niya dito na portion. Kaya ito yung resulting na cutted strand, no, ganun. But the thing is that there are some different types of enzymes, restriction enzymes that has been derived from different sources, different types of bacteria. Okay? So this is the applications of the restriction in the nucleus. So paano ba magamit ito? No? Applications of the restriction in the nucleus. So the restriction in the nucleus used in the field of genetic engineering to form a recombinant DNA or recombinant molecules of DNA, no? The compositions of DNA from different sources. Okay, so because of restriction enzyme, you will be able to cut a certain portions of the DNA, and that DNA, a portion of that DNA is desired traits. So i-cut natin yung desired DNA carrying the desired traits and transfer that into a host cell or host DNA para ma-change yung ma-modify ngayon yung characteristic ng host. In that case, yung host nagiging recombinant DNA. Okay? Okay, so same the restriction enzymes, vector and source DNA, some kind of sticky ends which can be joined by a ligase. So, yung mga sticky ends na naman na naputol, it can also be joined by a ligase, and it could be connected to the other ends of DNA. In which case, no, naputol yung strand ng isa at meron tayong naputol another strand ng another DNA, isampay natin yung DNA using the DNA ligase. Yan yan, no? So, mayroong illustration dito. Tingnan natin ito. Okay? So, actions of restriction enzyme. Basahin natin. So, the enzyme cuts both the DNA strands at the same site. So, for example, this is the vector DNA, no? So, mayroon tayong restriction enzyme. I-cut niya dito na portion. Okay? So, cut niya dito na portion. For example, this is from the, ang ginamit dito na enzyme is the ECOR1, no? Cut the DNA between the bases GT, no? Ito, i-cut niya dito sa between the bases G, guanine and adenine. So, tingnan natin. So, guanine and GNA. So, yeah. Sorry, dito pa pala tayo, no? The enzyme cuts both DNA strands at the same site. Ito naman dito, for example, cut niya dito sa GC, cut niya dito sa GC. So, dahil cut niya dito sa GC, so, ang mangyayari ngayon, ma-remove na itong, so, ito na ang resulting na pag-cut, ito na ang resulta, naputol na yung AATT, so, naputol na. Na dito sa kabila din naman, meron tayong isang DNA na na-cut din. So, ibig sabihin, pwede na sila mag-combine using a DNA ligase, no? DNA fragments joined by sticky ends. Itong sticky ends na ito dito, at saka itong sticky ends dito, mag-join sila using the DNA ligase. Ngayon, we have now a hybrid DNA. So, between the two, no? This is now the recombinant DNA. So because of the restriction in the nucleus, we're not able to perform recombinant DNA, no? Yun po ang application ng okay. So you have in here the diagrammatic representations of recombinant DNA technology, So we have the foreign DNA or desired DNA carrying the desired traits. So again, the same restriction enzymes cut both foreign DNA and a vector DNA. When we're talking about the vector DNA, we're talking about the plasmid, no? So, same restriction enzyme cutting both foreign DNA and the vector DNA at specific point. So, cut doon sa ating nucleotides, no? So, we have the vector DNA or the plasmid. So, tapos, itong plasmid, so, cut dito, mayroong cut portion using the restriction enzyme. And then we have the ligases joined foreign DNA to plasmid. So itong foreign DNA natin, itong the desired gene, ito yung foreign DNA natin, i-insert natin dito ngayon sa ating vector, which is the plasmid. So ligases join the foreign DNA. Then this is now the resulting plasmid carrying now the foreign DNA. For transformation, itong DNA na to, insert siya doon sa ating bacteria. Pag insert na sa bacteria, din ang cell ng bacteria mag-divide because that cell of bacteria carrying the plasmid carrying foreign DNA, then mag-divide siya hanggang ito siya will participate in the cell division using the cell division process of the host cells. Mayroon na tayo ngayong cells divide, meron tayo ngayong modified organism, no? This is the diagrammatic representations of recombinant DNA technology. So in short, meron tayong restriction enzymes which cut the portions of the plasmid para yung plasmid na yan, meron siyang puwang, meron siyang vacant portion. Yung vacant na portion na yan should be inserted with the foreign DNA, and so that the plasmid carrying now the desired traits from the foreign DNA, ang yung plasmid ngayon na carrying the desired DNA is to be incorporated in the host organism or the host cell, or it could be a bacteria, no? So that when that particular host bacteria replicates, so sasabay na ngayon sa replication yung plasmid natin carrying the foreign DNA from the different source. Okay? So that is how the recombinant DNA technology has been done. Okay, separation and isolations of DNA fragments. So the same, no? When you are to separate, when you are to isolate, of course, we have to use the tools of enzymes, no? And as well as other tools, no? So separations and isolations of DNA fragments. Okay. So actions of restriction enzymes result fragments of DNA. Now ngayon, because itong restriction enzyme cut the DNA strand into pieces at a certain portions of the nucleotides, so meron tayo ngayong mga pira-piraso or fragments of DNA because of that restriction enzyme. And so that meron tayong fragments, meron tayong particles of the DNA particles So we can separate now that the different fragments of DNA can be separated by gel electrophoresis, so that we can select, so that we can select what particular fragments of the DNA will be used in the biotechnology. Okay, and the separation isolations of DNA fragments depends on the negative nature of DNA. Okay, so separation depends on the natures of DNA. So we have the external electrical field is applied to the medium. So when we say the medium, so we're talking about, when we say the medium, we're talking about shall we say, we're talking about yung medium na nandoon yung fragments ng DNA. It has been cut into pieces by the restriction enzyme. Again, external electrical field is applied to the medium. I-apply dito sa fragments, no? Medium. Then, DNA moved to an anode. Okay, so when we're talking about an anode, anode actually is, we are talking about now in the charges. Now we have the cathode and anode, anions or ions. Because these fragments of DNA, they are charged particles. They will be able to attract into the charge. For example, the DNA applying the electrical fields attract to the anode or the negative side of the charge. Okay. The medium or the matrix use an agar gel natural polymer. No, itong medium na ito, ang gamitin nito use the medium or matrix use agar gel natural polymer extracted from the seaweed. Parang gelatin ito, no? Gamitin ito, parang gelatin. Okay? Use medium. Okay. So the medium now, the medium is contains the fragments of DNA. The medium now acts as sieve o parang gisala niya, no? Parang siever or separator. The medium now acts as sieve and separate the DNA according to their sizes, smaller fragments and further. So itong electrophoresis, gel electrophoresis, parang sala ito, no? Pagsasala ito ng different fragments of DNA separated by the actions of the restriction in the enzymes. The separated DNA, yung mga DNA na nag-attract doon sa anode portion ng charged particles, the separated DNA stained with ethidium bromide. So parang may ilagay doon na chemicals, itong mga separated DNA, stained with ethidium bromide and exposed to the ultraviolet radiations. Okay? Bright orange colored bands of DNA in gel. So meron siyang bright orange colored bands of DNA in gel doon sa medium ng agarose gel natural polymer. Okay? Then the separated bands of DNA now, itong bright orange colored bands of DNA, the separated bands of DNA cut out from the agarose gel and extracted from the gel phase is known as the elution. So we have now the elutions after staining. Then the purified DNA fragments, constructing recombinant DNA by joining them with cloning vector. So this is it, no? So mixtures of DNA molecules of different sizes doon sa medium under the electrophoresis process, no? Separation of DNA fragments on the sizes, parang salasala ito. Excuse me. Sorry pala. So kanina, ang anode pala is the positive side, cathode is negative side. Sige, source power. Ito yun, no? Ito yun na ang sinabi kanina na that the attracts to the anode or the positive sides. So electrophoresis, separation of DNA fragments based on size. Uh, so the fragments of DNA now will be separated based on their diameter or molecular sizes. Longer molecules and shorter molecules. Sala-sala ngayon. Then staining with ethidium bromide. Exposure to UV radiation, then cutting out DNA fragments. Yun yung ngayon ang process ng separation of DNA using the electrophoresis process. So yun po. So I think we have to stop up this portion, then we will continue our discussion next meeting on the cloning vectors. So thanks for listening, and so don't miss next time in our discussion, because if you're able to listen to our discussions, so you will easily understand the topics on this lecture. So thank you so much. Till next meeting.